So we'll start from here again today. And last day we just ended that it prefers glucose over lactose because in case of glucose, it can directly utilize the glucose. Glucose can enter the cell without any hiccups. But for lactose, an enzyme, particularly permease, is required for the E. coli to help in transport of the lactose from the exterior to the interior. Further, beta-galactosidase is required for the lactose to be broken down into glucose and galactose. Beta-galactosidase also plays a role in conversion of lactose to allolactose. So the preferred metabolite that that the E. coli uses is glucose, but in absence of glucose, it can use lactose. I can't see my options. Pen. Yeah. Now let's look into the Lac Operon. If we look into the Lac Operon, I think so we did this also yesterday. I gave you the basic glimpse of Lac Operon. This is the operon part. As you can see, this is the Lac gene. The Lac gene contains a promoter element, an operator element, the Z, Y, and A, 3 genes placed adjacent to each other, and a terminator element. And this is the Lac regulatory gene upstream of the Lac promoter gene, Lac operator gene. So if this is the Lac operator gene, we are having the Lac regulator gene here. Now you can see there is a promoter element. There is this Lac I gene, which is the Lac regulator or the Lac repressor, and the terminator region. This part of the operon is the polycistronic part. So the Lac operon is basically a polycistronic. It produces a polycistronic mRNA, and the Lac regulator gene produces an monocistronic mRNA. And Z produces the enzyme beta-galactosidase, required to convert lactose into allolactose. And also it helps in breakdown of lactose into glucose and galactose. The Y gene produces permease, which is essential for the lactose to diffuse across the E. coli cell membrane. And the A gene produces thiogalactoside transacetylase. Its function in lactose metabolism is not yet known. Now, what happens? When there is no lactose, what happens? So there are two conditions. In absence of lactose, what happens? And in presence of lactose, what happens? When there is no lactose in the system and E. coli is growing in presence of glucose, what happens? In normal condition, as you can see, it's written wild type. Wild type means in its normal condition, we see that the Lac I gene is a constitutive gene. So it constantly synthesizes the Lac repressor molecule. As you can see, it translates, the mRNA is translated, and then the mRNA is transcribed to produce, you can see the structures, the Lac repressor molecules. These repressor molecules have two domains, one with which they can bind to the DNA and one with which, if needed, they can bind with lactose, rather they bind with allolactose molecule. They do not bind with lactose molecule. They bind with allolactose molecule. So when the lactose is absent in the medium, Lac I gene constantly produces the Lac repressor molecule. And as you can see, the Lac repressor molecule goes and binds in the operator region. So the Lac repressor molecule has got itself attached to the operator region. Now, as we know that during transcription, the polymerase will identify and sit at the promoter. So the polymerase is sitting at the promoter, but on front of the promoter, you can see the Lac repressor molecules are sitting. So it becomes difficult for the polymerase to proceed further. So the polymerase cannot proceed further. Since this molecule is sitting, the polymerase cannot proceed further, and the polymerase opens up. So we do not get the transcription of the structural genes. Why are they called structural genes? Because each of these genes produces an enzyme. So it's producing a polypeptide, which is now having a different function. So the product of this gene is protein, and they're basically known as structural genes. So as we saw that since the polymerase cannot proceed further, because in the operator region, the repressor has been attached with the operator in a non-covalent interaction. So the polymerase opens up and there is no transcription of these downstream structural genes. And as you can see, we are not seeing any expression of these genes. So this is a situation that we get when there is absence of lactose. Now imagine if we put lactose into the system. The moment we put lactose into the system, somehow the lactose enters the cell. Now somehow we'll come to that later on. Lactose enters the cell. The lactose is converted to allolactose, and so the repressor molecules are produced. And in the system, in the E. coli system, since there is lactose, lactose has entered. Lactose has got converted to allolactose, and allolactose now binds with the repressor to inactivate them. In fact, if any repressor is also attached to this portion, allolactose goes and binds to this attached repressor also, changing its conformation so that it can no more remain in its bound form to the operator. And as a result, the lactose or repressor molecule opens up from the operator, and since the repressor molecule opens up from the operator, so now if RNA polymerase comes and recognizes the promoter region, it will not have any problem in synthesizing the downstream mRNA. So it skips the operator region and starts transcribing the mRNA from Z, Y, A. And as you can see, we are getting the mRNA. And since this is a prokaryotic system, what we are getting? We are seeing that translation and transcription are coupled. So the mRNA is hanging and the ribosomes have attached with the mRNA, and now they are synthesizing the three different proteins. From the Z gene, the permease is synthesized, galactosidase is synthesized. From the Y gene, the permease is synthesized, and from the A gene, the transacetylase is synthesized. So, since presence of lactose, with actually the presence of allolactose, opens up the repressor from the operator, and allolactose also, when binds with the repressor, brings about a conformational change in the repressor molecules, so that the repressor molecules can now no more get attached with the operator molecules, operator region of the DNA. As a result of this, we are seeing that the Lac operon is switched on. But now a vital question arises. If the operon is switched off in absence of lactose, then how come the first lactose molecule enters? And how come the first lactose molecule is converted to allolactose? So that is the question. Because as I said, in case of absence of, as you can see in this structure, if there is no lactose in the system, we are getting a situation like this where, just a second, let me erase all sides. We are getting a situation like this where you can see there is no production of the Z, Y, and A genes. So if there is no production of beta-galactosidase, permease, and transacetylase, then how come when the first lactose is given, that lactose enters? Because permease helps in entering of the lactose. And how come the lactose is converted to allolactose? Because beta-galactosidase helps in this conversion. So what happens when how come the first molecules of lactose enters if the operon is switched off? So this is the question. Now to answer this question, let us look into this particular binding region. The repressor is attached or bonded with the operator, as you can see. And I just mentioned that this interaction is a non-covalent interaction. So it is not a permanent binding. Obviously, it's not a permanent binding because when we saw in the next picture that allolactose binds to this molecule, automatically the repressor conformation changes and it can be released from the operator. So it is a non-covalent bonding. And in these cases, it is often seen that the repressor is not permanently attached to the Lac operator. At times, it opens up. And in this fraction of seconds, when it has opened up, again, I'm repeating, in this fraction of second, when it has opened up, if the polymerase at that given moment is sitting at the promoter, the operator has opened, the repressor has opened up for a fraction of a second, the polymerase shoots down in that time lag when the repressor has temporarily opened up from the operator. And if it can shoot down, it synthesizes one mRNA molecule, and that from one mRNA molecule, we can get one molecule of beta-galactosidase, one molecule of permease. That one molecule is enough. So the cytoplasm has a molecule of beta-galactosidase, a few molecules, 2 to 3 molecules of beta-galactosidase and permease present by the mechanism I just now said. And these few molecules will help the first lactose to enter the cell, and the first lactose to, and the conversion of the first lactose from lactose to allolactose. And once there is a conversion of one molecule of lactose to allolactose, then it becomes very simple that allolactose goes and binds with the repressor molecule now attached with the operator, changes its conformation, the repressor opens up from the operator. So now polymerase is free, and this results in a very enhanced, increased rate of transcription of the Lac Operon. So as you can see, in the presence of lactose and the absence of glucose, just now we discussed, the amount of each enzyme increases coordinately or simultaneously by the mechanism I just now told, about 1,000-fold. Because the 3 essentially inactive genes are now activated and are actively transcribed. This process is called as coordinate induction. So we see a phenomena of coordinate induction in Lac Operon. So we realized that how the first molecules of lactose can be entered within a cell by few molecules of the enzymes that has been already produced. And once the first lactose is converted to allolactose, allolactose efficiently removes the repressor from the operator region, and then the transcription of that particular mRNA increases many-fold. Now, let us look into certain situations of the Lac Operon. Now, let us come to a condition where we are seeing that within the E. coli cell, there are 2 sets of Lac gene. So this is one Lac gene, and this is another Lac gene. Okay, and we are having the repressor gene. You can see this is the repressor gene in one chromosome, and this is the repressor gene in another chromosome. Okay, just give me 1 second. So as I was saying, so we are having 2 sets of Lac gene on the chromosomes, and you can see that this is the Lac repressor gene producing the repressor protein. This is the Lac I gene producing the repressor protein. Now look at this DNA and look at this DNA. Now we are seeing that this is the operator DNA. The sequences in the operator DNA are normal. There is no mutation. Mutation means there is no change in sequences of the DNA present in the operator segment. As a result, the repressor can go and bind with the operator region. But here we are seeing that there is some change in this operator sequence. So there is a mutation. Some mutation has come up in the operator sequences. As a result of this mutation, when this repressor comes and binds with the operator, it fails to bind with the operator because the sequences have changed. Binding of the operator protein to the DNA segment is very specific, and the sequences need to be the same. But here, since there is a mutation in this operator sequence, we can see that the repressor cannot bind to this operator sequence. Now, this is a condition when there is no lactose in the cell. So if there is no lactose in the cell, and as you can see, we have one DNA with a normal operator and one DNA with a mutated operator. Lac I plus gene is normal. This is the repressor gene. So we are having the normal repressor protein. The normal repressor protein can bind with the normal operator, but it fails to bind with the mutated operator. Now, in this case, as we see, polymerase can come and sit here in this promoter. Polymerase can also come and sit here in this promoter region. In this case, as the polymerase tends to proceed forward, it finds a block in this area. But in this case, as the polymerase tends to proceed forward, there is no block. So the polymerase can slide this part and starts transcribing the Lac metabolizing gene mRNA. So it is transcribing the Z, Y, and A mRNA to produce the beta-galactosidase, the permease, and the trans-acetylase enzyme. So even though lactose is not present, you can see on the upper half, the gene is switched off, no transcription. But in the lower half, since the operator is itself mutated and fails to bind with the repressor, we are seeing that the Z, Y, and A genes are transcribed. So the lower half is switched on. So for the lower segment, for the lower DNA, we can say that it becomes irrelevant whether lactose is present or not. This particular DNA will be constantly switched on. It becomes totally irrelevant if lactose is present or not. Because in absence of lactose, you can see the enzymes are produced. In presence of lactose, as I showed you, the enzymes will always be produced. So it is irrelevant now that lactose is there in the system or absent from the system. The enzymes are produced. Hence, it is known as constitutive synthesis. So operator has lost its control. Since operator has got itself mutated, it has lost its control on the Z, Y, and A gene. And since there is no control of the operator on the Z, Y, and A gene, the synthesis is known as constitutive synthesis. There is one more point to be mentioned here, is that when the operator loses control or when the operator is itself mutated, as you can see in the picture, we are seeing two DNA. In one DNA, the operator is normal. In the other DNA, the operator is mutated. When we look at the two DNAs, what are we seeing? The one with the normal operator, the lac is behaving normally. So in absence of lactose, there is no mRNA production. But when the operator is mutated, you can see the operator has lost control over Z, Y, and A. In absence of lactose, also we are seeing the production of the three enzymes. So such kind of DNA sequences which exerts a control on the genes sitting downstream or adjacent to it are known as Cyst dominant gene. So the operator sequences are said to be Cyst dominant. That means it is actually controlling the production of the genes sitting on the DNA on which it is. So if this is the operator is sitting on this DNA, it is controlling the production of Z, Y, and A. It cannot control the production of Z, sorry, it cannot control the production of Z, Y, and A on the other DNA. It cannot control this. It does not have any control on Z, Y, and A of the other DNA. It can only has an effect on the Z, Y, and A of the DNA on which it is sitting. So you can see this is called Cyst position. Lac Z, the blue color Lac Z is sitting in adjacent to OC. So it is in Cyst position to OC. Hence, the operator is known as cis dominant in nature. So just a second. So we discussed about this operator mutation, and we saw that, as I just now said, that operator is a cis dominant gene, and this mutation is known as OC because once we have this kind of mutation, the operator, I mean to say the Lac Operon is becoming constitutive in nature. We are seeing that the operon is now constantly switched on even though lactose is absent. So, and this particular mutation, OC, operator constitutive mutation, is dominant to the normal gene O+. As you can see, on the other side there was O+, and on this side there is OC. But if you look at the output, we are seeing that in absence of lactose, we are getting the production of all the three genes. If we look at the output as a whole, consider this system as a whole system, then we are seeing that the operon is active. Just a second. So as a whole, when you consider this system, we are seeing that there is production of the three enzymes in absence of lactose. So we have to consider looking at the two DNA as a whole. Even though we are seeing that in one DNA there is no production in absence of lactose, in another DNA there is a production of the three enzymes in absence of lactose. But from a scientist's perspective, we are looking at how the cell is behaving. We are not looking as to how the two individual DNA are behaving. So if we look into the cellular mechanism from outside, what we are seeing that in absence of lactose also, the three enzymes are being produced. And that means that this particular mutation is dominant to the wild type gene. O+ is the wild type gene. OC is the mutant gene. OC becomes dominant to O+ because we can see as a whole, when we consider the picture, we are seeing that we are getting the production of the three enzymes. So it's switched on, plus. Now let's come to another picture where you can see the similar picture, the one that I just now showed you in absence of lactose. Now if lactose is given, what happens? Lactose is converted to allolactose. Allolactose efficiently removes this repressor. And in this case, as we saw, in absence of lactose, this was switched on. In presence of lactose also, this was switched on. Okay? But in presence of lactose, once we remove this repressor molecule, this is now also switched on. So in presence of lactose also, from the outside perspective, what we are seeing that the operon is switched on. That is normal. In presence of lactose also it is switched on. But what is the difference? In this case, we are getting more enzymes produced because both the DNAs are now producing the enzyme. So the amount of enzyme that is produced is doubled as respect to the previous one, where the amount of, this is the amount, this DNA is only producing the enzyme. So with respect to this scenario, when we look into the next one, we are seeing that in presence of lactose, the amount of the enzyme produced has doubled. So when the amount of so the amount of the enzyme has doubled. And if we look into the interior of the cell, now if we look at the DNA structure, we saw that in the previous slide, the lower DNA was on, it produced its enzymes. Here also the lower DNA is on, it is producing its enzymes. But in the upper DNA, we saw in the previous slide, there was no production of the enzymes because lactose was not there. But now because there is lactose, there is this production of enzymes. So lactose has induced or switched on this DNA. And hence the synthesis is known as inducible synthesis. And lactose does not have any control on this DNA. Hence this synthesis is now known as constitutive synthesis. And as I again said, once more repeating, that this segment of DNA sequence does not have any control over this DNA. It can only exert its effects on the sequences which are downstream to it. So it's basically a cis dominant one. So operator mutations are cis dominant one, and OC is dominant to O plus. I'd like to give you one more concept here. If you look into this particular picture, pointer option series, let's get back down to the other picture. Let's look into this picture. Here, in absence of lactose, we are not getting any production of mRNA. There is no mRNA produced. Only in presence of lactose, we are getting the production of an mRNA. So lactose, or rather, the allolactose is the inducer for this operon. And the operon is said to be an inducible operon. So we have seen here is a case of an inducible operon. So in absence of lactose, no mRNA. In presence of lactose, mRNA produced. Hence, lactose is the inducer molecule. In presence of lactose, the Lac Operon genes are transcribed. So that was the first category of mutations we studied. That was operator mutation. And we understood that the operator mutation, OC, is dominant to the O plus wild type gene. And the operator mutation is a cis dominant one. The operator gene is in fact a cis dominant one. The operator mutation can control the production of the structural genes of the DNA on which it is sitting. It does not have any control on the DNA present at the opposite end. So OC can control the Z, Y, and A expression of the DNA on which the OC is sitting. Now we are coming to another category of mutation, that is the mutation of the repressor gene. So now we are moving on to the repressor gene. Concentrate on the repressor gene. This is one DNA that has been shown. And this is the I minus mutation. As you can see, this gene is producing a repressor protein, but the proteins, as the gene itself is mutated, the proteins are defective. And when we studied the operator mutation, one thing was very clear from the fact, from that information that we got there is that the repressor and the operator binding is very specific. A single change in a nucleotide can affect the binding. So similarly, when we have a defective repressor protein, this protein will not be able to bind with the operator sequences. It will not be able to identify its binding sequences. So since this binding is not occurring, as a result, what is the net scenario? The net scenario is that in absence of lactose, let's look at here. So lactose is the inducer. So in absence of lactose, allolactose, the repressor gene has been produced. The repressor gene is active but mutated. Defective proteins are being produced, but they're incapable of binding to the operator region. Since they're incapable of binding to the operator region, so whenever the polymerase is sitting, the polymerase can easily transcribe the Z, Y, and A gene. Since the polymerase can easily transcribe the Z, Y, and A gene, we are constantly getting the production of beta-galactosidase, beta-permease, and trans-acetylase. So as you can see, in this scenario also, the operon has become constitutive. Lactose is not exerting any control on the operon. The operon is automatically switched on. It does not need lactose to switch it on. So this is another situation. This is another mutation where we are seeing that the Lac repressor mutant proteins cannot recognize the operator, and as a result, the Lac gene are again constantly transcribed. So this is a scenario, this is a picture where I am showing you a single DNA strand. Now let's imagine what will happen if there are two DNA strands. This is interesting. In this particular case, the normal allele is I plus and the defective allele is I minus. So this is how you show the chromosomal scenario. So on one DNA I plus is sitting, on the other DNA I minus is sitting. And for information, I plus is dominant to I minus. If this is the scenario, now see, in absence of lactose, what happens? We are seeing the production of normal repressor from the I plus gene, the production of defective repressor from the I minus gene. But as I told you, I plus is dominant to I minus. So the protein products of the I plus gene is dominant. So these protein can go and bind to this operator as well as this operator. Even though this repressor gene is defective, see, this repressor gene is defective. It is producing a defective protein. But this repressor gene is normal. It is producing a normal protein. And this normal protein is capable of binding to both the operator. It can bind to the operator on which the normal gene is, repressor gene is sitting, and it can bind to the operator on which the defective repressor gene is sitting. So this particular repressor gene can exert its effect on the operator adjacent and downstream to it, and also on the operator sitting on the opposite DNA. So repressor is known as transdominant because one repressor gene, if the need requires, can exert its effect on both the operators. Here I plus can exert its effect because, as I said, the I plus is dominant to I minus. So whatever I plus will produce is the dominant protein, is a dominant molecule, and hence it will go and bind to both the operators. So in absence of lactose, so we get a scenario similar to the normal one. No product. So what do we see overall? Minus. Lac operon is switched off. No production of the enzymes. But when lactose is given, now lactose is given. Now, since the repressor molecules had got attached to both the operators, as we saw in the earlier slide, that this repressor molecule had got attached here and as well as here. So when we give in lactose, what happens? The lactose is converted to allolactose, and the repressor molecules are removed from this operator as well as this operator. And now both the strands of DNA are capable of synthesizing or transcribing the mRNA and translating it into protein. So both the DNA strands are capable of synthesizing the required structural genes. So we are getting inducible synthesis in both cases. So this is one thing that should be kept in mind, is that the operator gene is basically a gene or a certain amount, a stretch of DNA containing some sequences. The repressor gene actually has a protein product. The total control of the operon is actually maintained by the binding of the DNA sequences of the operator with the repressor protein. If there is a mutation in the operator DNA sequences, it will have its effect only on the gene sitting adjacent to it. But if there is a defect in the repressor gene, then the protein becomes defective. If we have a normal repressor on the other DNA, and if it is producing a normal repressor protein, this normal repressor protein can bind to both the operators on the DNA, because the repressor proteins are available in the cytoplasm, so they can move to both the DNAs. And hence the repressor is a transdominant one, because the repressor has a protein product. It is controlling the operator via the protein product. So it can control both the DNA if it requires. Hence it is a transdominant in nature. But in case of the operator molecule, it is only DNA sequences. It does not have any protein product. So these DNA sequences cannot move to the other strand. So they can only control the genes sitting on the same strand. They cannot control any genes on the other strand. Hence, it is cis dominant. So whenever a gene is producing a protein and is exerting its control through the protein product, usually such genes are basically transdominant in nature. I've again lost my count. So basically these genes are transdominant in nature. So we studied the effect of I plus and I minus mutants. Now let us look at another mutation that is observed in the repressor gene, which is known as IS. And let me be very clear, IS is dominant to I plus being the wild type gene. So we are having a normal Lac gene and a IS and a repressor gene having a IS mutation. So this is the normal Lac repressor. This is the mutant Lac repressor. The mutant Lac repressor is producing defective protein, and the normal Lac repressor is producing the normal protein. But you can see the scenario, the defective is dominant to the wild type. Here, since IS is dominant to I plus, this defective protein goes and almost binds permanently to both the operator. As it permanently binds to both the operator, so in absence of lactose, the defective repressor is attached to the operator, both the operators. And in presence of lactose also, the defective gene is attached to both the operators. So this defective gene is super repressor. It is totally shutting off the Lac operon. So in absence of lactose, the operon is switched off, fine. But when there is inducer molecules present, allolactose is present, in spite of that, or if there's lactose molecules present, in spite of that, it is almost shutting off the Lac operator. As I said, this is not a covalent bonding. So for fraction of seconds, if by chance it opens, then there will be very few molecules of the three enzymes produced. So the allolactose will enter. But allolactose cannot, the super repressor molecules cannot recognize this allolactose. So they do not open up from the operator. They remain attached to the operator. So if they constantly remain attached to the operator, then very few molecules of the mRNA will be produced, as compared to the fact that had they had opened up from the operator, then the transcription rate would have increased almost 3,000 times. So that doesn't happen. The super repressor permanently in a non-covalent interaction remains bonded with the operator. And thus, in absence of lactose, the operon is switched off. And if you look into this scenario, in presence of lactose also, the operon is switched off. So this is the second repressor mutation. And there's a third category of repressor mutation, which I don't have a picture for that. So I'm just explaining it here. Just a second, let me pointer options erase. The thing is that there's a third category of repressor mutation, I minus D, which is dominant to I plus. Now, when we look at the repressor protein, the I plus is producing a repressor protein, which is a tetramer. So let us imagine there are 4 red balls. This tetramer actually recognizes the operator sequences and switches off the operon in absence of lactose. And in presence of lactose, it opens up from the operator and in absence of lactose, switches off the operon. And in presence of lactose, it binds with the allolactose and opens up from the operator. Now, let us look at I minus D. I minus D is a defective repressor protein. And let me change the color of the pen just a second. Give me. Now, this defective repressor protein is again producing a protein with 4 tetramers that, as I said, it is made up of 4 subunits. What happens? I plus gene is producing 4 red color balls, as you can see in the picture, and I minus D is a defective one, which is producing 4 orange color balls. Now, these polypeptides are produced in the cytoplasm. It is often seen that the 2 orange balls, where did my cursor go? The 2 orange color balls, sorry, pointer options is so I'm having 2 orange color balls, and now let me change the ink color. Red. Join up with 2 orange color balls. So these proteins are also produced in the cytoplasm. These are also produced in the cytoplasm. But when a scenario like this happens, 2 orange color joins with 2 orange color joins with 2 red color balls to form a defective tetramer. So when this kind of defective tetramer is formed, so the repressor protein is again defective. It fails to recognize the operator. So in absence of lactose, this defective tetramer will not recognize the operator. So automatically the operon will be switched on. And in presence of lactose also irrelevant. It is not bonded with the operator. So the operon will be switched on. So this is the third category of mutation of the repressor protein. So when we have a scenario like this, আমরা operon যেটা পাবো, operonটা permanently switched on. So the main discovery of the Lac Operon and the mechanism by which it was done was provided by Jacob and Monod. So these are the 2 scientists who actually studied the regulation of expression of the Lac Operon genes. This is the Lac Operon organization. And we have the Lac Operon, as you can see, the ZYA gene. This is the promoter element, operator element. So as you can see, the RNA polymerase sits on the promoter and transcribes the ZYA, which is translated, this is a polycistronic mRNA, which is translated into the three enzymes, galactosidase, permease, transacetylase. Permease helps in entering of the lactose molecule. Beta-galactosidase helps in breakdown of the lactose to glucose and galactose. Now coming to the Lac operator. We saw is basically a DNA sequence which is sitting adjacent to the promoter. And now we have the Lac regulatory gene. So a polymerase sits on the promoter, produces the Lac repressor mRNA, and we get the Lac repressor molecules. So this is the basic scenario of the Lac Operon. Now, in absence of lactose, just as I said, you see the repressor is binding. So even though the polymerase sits, it cannot proceed. The moment there is lactose in the system, converted to allolactose, the allolactose will now go and bind with the repressor, conformational change, and the repressor opens up. So the polymerase sits and transcribes the mRNA. So with this, tomorrow we'll take up with the next part of the mutations. We have yet to study the Z, Y, and A mutations.